We're going to diagram DNA structure. DNA can begin with what is known as the double helix. Make an S, make a slightly lower S, and then connect them. This allows you to label another feature of the double helix, which would be the minor and the major groove. And of course, we need to build this polymer out of monomers. So we are going to start by constructing a nucleotide. The nucleotide has a few different parts. This portion is called the nitrogenous base. This is the pentose sugar, and this is a phosphate. It's important to note that there used to be 3 phosphates on here, but 2 of them have been removed from a nucleoside triphosphate. DNA acts as its own TP as it is being assembled. And so, noting the connections that need to happen between these nucleotides, you can place another one below it, and then connect what is ultimately going to be carbon 1, 2, 3 to carbon 4, and then the 5th one is up here, so we are making 3 to 5 connections. And ultimately what you're going to want to do is have a few different nucleotides. I'm just going to stick to 6 here. Now on the other side, the nucleotides are upside down. They're flipped 180 degrees. So if it helps to flip your paper around to do this, you can certainly do that on a test or a quiz. Or if you want to just imagine them upside down, that can help as well. Don't forget the 3 to 5 connections. And we can start labeling the structural features here. I will simply highlight them for you. First of all, we've got a nucleotide, which we can circle and symbolize right there. Nucleotides are connected to each other with hydrogen bonding. There should be 2 hydrogen bonds for A, T, 3 hydrogen bonds for G and C. In addition, we can label the 3 and 5' ends. The 5' end is where the 5th carbon is. The 3' end is where the 3rd carbon is. This creates a direction that the DNA will ultimately be read, where this side goes from 5 to 3, 5 to 3, which means these 2 strands are anti-parallel. In addition to that, we can highlight the sugar phosphate backbone on one side. We can also highlight the base pairs connected by hydrogen bonds. And then the last thing to note is that one of these strands is going to represent the actual DNA code. We can call that the coding or the sense strand. And then the opposite strand would then become the non-coding or the anti-sense strand. Here's a full diagram that's a little bit neater for your studying pleasure.